Should you stop or should you go? Well, if you're a cell, it's all about the signals. Hi, biology friends. It's Sarah, your other biology professor, and let's talk about cell cycle checkpoints. In this video, we'll talk about why the cell cycle has checkpoints, and we'll take a little tour of 3 of the major cell cycle checkpoints, seeing why a cell might stop at that checkpoint and some of the key molecules involved. Now, in another video, we saw how molecules acted as positive regulators of the cell cycle. Phase-specific cyclins activate cyclin-dependent kinases that phosphorylate target proteins that work to move the cell cycle forward from one phase to the next. And if everything worked perfectly all the time, that'd be all you need. But sometimes we need to hit the brakes on the cell cycle to arrest the cell cycle, to stop moving forward. Why? Well, it could be that it's just not the right environment for the cell to divide right now. Or maybe that cell has damage. And we don't want damaged cells to divide and produce more damaged daughter cells. And so we've got molecules acting as negative regulators of the cell cycle. And you'll see these guys acting at cell cycle checkpoints. A cell cycle checkpoint is basically a quality control mechanism where the cell is going to do a bit of an assessment and decide whether it's appropriate to divide or not. So just like at a factory where a product has certain standards it has to meet before it can get its inspected by number 43 or whatever sticker and be shipped out, this cell also has a list of requirements that it needs to meet in order for the cell cycle to continue. Now, there are many checkpoints throughout the cell cycle. Quality control is always watching, but here are the 3 major checkpoints that you are likely to run into in your class. The first checkpoint is often considered the most important, and you'll find that toward the end of G1. What happens in G1? Well, the cell is growing and it's preparing for the next stage where we're going to replicate the DNA. And that's why this checkpoint is a biggie. Because once the cell moves on from here and replicates its DNA, it's essentially committing to divide unless major problems pop up later, because it's doubled its DNA. But in order to pass through this G1 checkpoint, this cell needs to run through its checklist. And at this checkpoint, we're assessing things like, are there signals to move forward? We're talking growth factors here. Growth factors are going to stimulate production of cyclins, which activate cyclin-dependent kinases, which are going to trigger production of the proteins needed for the next step. Cell size. If that cell is going to divide and become 2 cells, it's going to need to meet certain size requirements first. Nutrient availability. Division is a very energy-intensive process, so the cell needs to make sure it has enough energy reserves and materials in order to get through that. And likely the most important, DNA quality. Remember, the next step is DNA replication, and we don't want to replicate damaged DNA. If the cell can't check all of those boxes, it shouldn't be moving forward. Well, at least not yet. A cell that doesn't pass the checkpoint can get put in a sort of cell cycle timeout, a pause, until its problems are resolved. And so when the cell wants to take a little pause in response to some fixable DNA damage, it's going to increase production of a protein called P53. P53 is a transcription factor. That means it influences the rate of transcription for a variety of genes. It's going to influence what and how much product the cell is making. And one of the products that P53 promotes the expression of is cyclin-dependent kinase inhibitors. Remember that cyclin CDK complexes are one of our go signals to push the cell cycle forward. But P53 says, yeah, none of that right now by stimulating the production of inhibitors of those kinases, basically shutting them down and stopping the cell cycle. Depending on the type of DNA damage, different repair pathways with different proteins involved will be activated. So at any point in the cell cycle, double-stranded DNA breaks, where the helix has broken in the same place on both strands, might be responded to by proteins like BRCA1. And you might recognize that name since mutations in the BRCA genes are associated with a significantly increased risk of many cancers, including breast cancer. Now, if a cell gets the all clear and is allowed to progress through that G1 checkpoint, It'll copy its DNA during the S phase and meet the next major checkpoint in G2, the last stage of interphase. So what's on our checklist at this point? Well, as always, we're looking for DNA damage, and we already know what happens if we find that. But we're also looking to make sure that all of the DNA was properly replicated. We don't want to go into mitosis if DNA replication isn't complete. So here again, the cell cycle can be paused while the problem is fixed, either by repairing the DNA damage or completing DNA replication. A cell that is cleared to move on will find the next major checkpoint during mitosis, the M checkpoint, specifically during metaphase of mitosis. What happens during metaphase? The chromosomes will be lined up in the center of the cell, and that's due to the push and pull of the spindle fibers aligning them on that metaphase plate. In the next phase, anaphase, sister chromatids will be pulled apart. And so at this metaphase checkpoint, the cell is looking to make sure that all of the chromosomes are properly connected to the spindle through the chromosome's kinetochores, the proteins along the centromeres. And that's why this checkpoint is also called the Spindle Assembly Checkpoint. And it's important because if a chromosome is not properly attached to the spindle and that cell goes on to divide, we're going to get daughter cells with missing or extra chromosomes. That's called aneuploidy, and it can cause some pretty big problems for the cell. So if there are issues with spindle attachment, we don't want to move forward into anaphase. The proteins that hold the sister chromatids together will stay intact until that problem is resolved. At each checkpoint, we've mentioned pausing or arresting the cell cycle until problems can be fixed. But what if they can't be fixed right now? For an extended cell cycle timeout, cells can exit the cell cycle into a non-dividing stage called G0, either temporarily if conditions improve or permanently. When damage is unrepairable, P53, P53 again, triggers apoptosis, programmed cell death. Basically, destroy that damaged cell so it doesn't cause any problems for the organism. We remember P53 is a transcription factor, and so it's going to induce transcription of a lot of different genes needed for the apoptotic pathways. And all of these broken up cellular components will be shuttled into little apoptotic bodies called blebs that bleb off of the apoptotic cell and will be engulfed by the immune system. Genes encoding proteins like P53 that inhibit the cell cycle or induce apoptosis are called tumor suppressor genes because they inhibit the sort of unchecked growth that could lead to cancer. Mutations in tumor suppressor genes that produce dysfunctional products could result in damaged cells being able to pass through checkpoints that they shouldn't. But we'll have to look at that another time. For now, give me a thumbs up if you learned something about cell checkpoints and the negative regulators of the cell cycle. Check out my website for tutoring resources, subscribe for more biology content from me, and I'll see you next time. Bye.